This video is on X inactivation, specifically dealing with the example of tortoiseshell cats. The Lyon hypothesis was proposed by Mary Lyon. She claimed that if you have 2 or more X chromosomes, one will be randomly inactivated due to X inactivation. The spot where the inactivation occurs is called the Barr body. In this picture, you can see the 2 X chromosomes, one from both mom and dad, and you can see how they undergo a random inactivation, producing one active X chromosome and one inactive X chromosome. The spot up here where the inactivation took place is called the Barr body. So these are all of our projected genotype and phenotypes for black, yellow, and tortoiseshell cats. You can see the males can only be black and yellow, whereas females can either be black, yellow, or tortoiseshell. These squiggly circles are there to represent the inactive X chromosome. When looking at the 2 tortoiseshell cats, you can see how they both start off with a heterozygous mix of both black and yellow fur colors. Then one of the X chromosomes becomes partially inactivated, producing 2 different colored patchy fur colors. This is the example of the cross between a yellow short-haired female and a black long-haired heterozygous male. The female's genotype is little h, X little b, X little b. The male's genotype is big H, X, big B, Y. To find the cross results, you can either do a Punnett square or a phenotype branch diagram. And these were the results. So half of the results were male and half of the results were female. And from the female, half were short and half were long, and same with the male. The female, the half short and half long, were 100% tortoiseshell, and the male, half short, half long, were all 100% yellow. And that resulted in 1/4 short tortoiseshell, 1/4 long tortoiseshell for the female, and then for the male, 1/4 short yellow and 1/4 long yellow. So why are the coat colors on the tortoiseshells different colors? The patchiness occurs because of an X chromosome is inactivated in a cell. Most of its surrounding cells have the same X chromosome inactivated, which produces the patchy pattern for the expression of an X-linked characteristic found in heterozygous female cats. The mix of these black and yellow patches comes from one of the X chromosomes not being fully inactivated. The black patches are clones of cells derived from the original cell, where the orange was the inactivated X chromosome. The orange patches come from the original cell where the black X chromosome was inactivated. You will never see the same tortoiseshell cat colors with the same type of patches due to this. In this particular cross, the tortoiseshell color would depend on if the black or yellow X chromosome was the one not fully inactivated and what the neighboring cells looked like and which of the X chromosomes were inactivated.